Okay, so did you guys have any questions on the exam 3 material that we finished up or from that recorded lecture? No, you can ask me after if you want. Send me an email. Come talk to me. Okay. So we're moving on to chapter 4 material. The nice thing is this actually will be a little bit of a review of some of the stuff that you've learned for exam 3, and which is nice because, as I told you guys before, get that muscle physiology under your belt because we're adding to it. So the nervous system and nervous tissue, chapter 12, we're going to look at how it's broken down, the various divisions and its functions, and this is based on what they can do for us. So if you guys remember when we talked about positive and negative feedback mechanisms for homeostatic balance way back, like exam 1 material, do you guys remember that a little bit? So we use the example of a thermostat, for example, right? So you have a receptor, a control center, and an effector. Okay, so we're going to use and apply that terminology here to sensory, integrations, and motor. So sensory, it says, is monitor changes in internal and external environment. So it's changing in response to, it's getting information. So do you guys think that that would be a receptor, a control center, or an effector? If it's sensory. Sensory would be a receptor. So we have tons of receptors on our body. For example, like when you touch stuff and you get a sensation, something's cold, something's hot, something's hard, something's soft. You are just gathering information. And where do you think you send that information? We have to send it to something that can process it and understand it, right? To the brain, which is going to be our control center. So integrations are processes, interprets, and decides regarding the sensory input. Does that, if you think about some of those words, it kind of makes sense that that would be your brain has to decide what that information means and what do you want to do about it. So if something is cold, maybe that's not a big deal, but maybe your brain says, oh, I don't want to touch something that's cold right now. Or maybe something is hot, not so hot that you burn yourself, but you're thinking, well, that is hot. Should I hang on or should I let go of this, right? So I'm going to put for sensory, that was our input, if I can. So input, things that are coming in, information coming into our body, which if we use the other terminology is a receptor. Okay. Integration then we're saying is we're trying to decide what to do with this information. So this is our CNS or central nervous system, and our brain is part of that. And this is what we're going to call as our control center, which makes sense. And then for motor, what do you guys think motor is? So it says it's response pathways that activate glands and muscles. So maybe if something that we were touching is a little too warm for us, we think it's a little hot, and we don't want to hold on to it anymore, our brain decides that we don't want to do anything with it, and it tells our muscles to let go and don't hang on to it, right? So these would be the effectors, which is the output. So input means information coming into your body. Your central nervous system or control center has to decide what that information means and what should we do about it. And then the effectors or motor is going to be the output, the information that goes from our brain out to the other parts of our body that decides what should we do now because of that information. Okay, so sensory doesn't do anything with it. It just takes the info to the brain and tells the brain what's going on. So think of it just like the thermostat, okay? Another way of thinking of sensory or input is if this information is coming into our brain, okay? Another term would be ascending or afferent. So you can think, so we'll put, these are terms that you're going to see in a little bit some more. So ascending, I kind of think of it going up to the brain, okay? And afferent also means coming to the brain. So kind of like with the muscle physiology, you guys had to learn some new terminology. Same thing with this, but hopefully some of it will be a review and it won't be too new. For motor or output, the effectors, another way to think of that would be descending, because think of it as going from the brain down to the parts of your body that can do something about it. So descending. And another term would be efferent. So you can think efferent, E for exit. So think it's exiting the brain to send the information to tell you what else you need to do. Move those muscles, tell your gland to secrete something so that you can have a certain hormone or action occur. Okay. So if we look down at that picture and we start looking at levels of organization, there is structural and there is functional. Before we talk about structural versus functional, let's talk about what CNS and PNS means. CNS is central nervous system, and that means that it's just the brain and the spinal cord, nothing else. That is your CNS. If you're talking about peripheral nervous system, or PNS, that is talking about nerves and ganglia. So if something is called the sciatic nerve, you automatically know you're talking about peripheral nervous system. If we say it's cranial nerve number 3, is that CNS or PNS, do you think? PNS, because it's a nerve. Even though cranial nerve is telling you it's coming directly out of the brain to these other areas, it still says nerve. It is still a PNS then. It is not the brain itself. It is not the spinal cord. So CNS is only brain and spinal cord, and then everything else has to be PNS if it's then the nerves and the ganglia. Okay. Let's see, if we break it down a little bit more to protections for the CNS, is that down too low? No, histology. If you guys think about protection for your central nervous system, what protects your brain and your spinal cord? Your skull and your spinal column, which you can think of as like armor, right? So your brain and your spinal cord are very soft. You guys have your brain lab this week, correct? Yes. And so you guys will see how soft, even though it's a preserved sheet brain, you'll see how soft and mushy it is, and why it's good that we have something so hard like our armor of the skull and the spinal column to protect it. Now, what about the nerves, the PNS side of it? Does that have bones, like a bony column armor protecting it? So what the PNS has protecting it is really what we call CT wrappers, connective tissue wrappers. So things that can insulate and help protect it, but overall, those guys are not well protected, not like our brain and the spinal cord are. Okay. So the two main subdivisions of the PNS are sensory and motor. So notice here is sensory, and there's that term afferent, and motor has the term efferent. It's referring back to what we just saw a minute ago, okay? So with sensory, afferent means receptors are sending information to the CNS, right? So to CNS, info to CNS, okay? And it's input information, because it's coming in, information that you're getting coming in, and then we said it's also ascending, right? I know it's kind of repeating myself, but it's nice to repeat it when it's new information. As far as for motor or efferent, we said E was for exit, so we know that this is going information from the CNS. So from CNS, bless you. And it's a response to the effectors, and the effectors are going to be your skeletal muscle and your glands. And so this is output. And it's descending because it's coming out from the brain to those other areas, away from the brain. Okay. As far as functions and, no, that was the beginning of this page. All right. It's so quiet in here. So when we do sensory afferent division, info to the brain, you also have two more divisions within that. So you have somatic, soma meaning body, okay? And visceral, have you guys heard the term visceral before? Visceral means organ, right? So somatic means it's coming from the skin, the muscles, the joints, somewhere about your body that it's telling your brain information that's going on, versus if it's visceral information, it means it's coming from the organs. And have you guys ever had, if anybody's had like appendicitis or kidney stone or something going on in their intestines or their stomach, And did you notice how the information isn't really obvious and diffuse? I mean, it is diffuse. It's kind of spread out and you don't really know what's wrong. You just know your belly kind of hurts sometimes. So visceral information is not always super specific in the beginning. And the longer it goes on, the more it hones into the spot that's really a problem. So if you had appendicitis, like the whole abdomen kind of hurt, and then maybe a couple days later, it started to be more like the lower right quadrant that was hurting. Versus if you have information somatic from the skin, muscles, or joint, you know if you have pressed up or touched something. It's a little bit more obvious. It's a little more non-diffuse. Okay, so it's a little more obvious to me when you're talking about the body. As far as the motor division, so efferent information from the CNS, output going out to the muscles or the glands. And so for here we have somatic nervous system versus autonomic nervous system. So somatic would mean like your skeletal muscles. Okay, so you're telling your muscles to move and do something. So again, soma, think body, think muscles are going to move your body. Autonomic, think more like automatic. You don't have to think about it. Your body's going to take care of it. It's going to do it automatically, kind of like autopilot, okay? So autonomic, think like your cardiac muscle, your smooth muscle, and your glands. You aren't telling your heart to do things. You're not telling your esophagus to push the food down. You're not telling your stomach how to digest. You're not telling your bladder, go now. I mean, you can tell it not to go now, right? You learn to teach it that. And glands, you don't sit there and go, all right, salivary gland, work. No, I mean, you could think about food and automatically you get a response and start to salivate sometimes, but these are autonomic, you're not really controlling it. So that's why it says controls cardiac, smooth, and glands. Now, of the autonomic, you have sympathetic versus parasympathetic. So you'll see me abbreviate sympathetic with an S and parasympathetic like P/S, okay? So sympathetic, or the sympathetic nervous system branch, you could think of it as sympathetic to your survival. This is fight or flight, okay? So this makes sense, right? You guys have heard about fight or flight? You either decide to fight off whatever is going to hurt you, or you run away from it so you can survive another day, right? Versus parasympathetic is more of a rest, digest area. Rest, digest. And I know it seems silly that we're using things that rhyme, but it kind of helps you remember it. So most of the time, your body is in rest, digest mode. You're sitting here listening to me talk, it's rest, digest. If I said, pop quiz right now, you might go into sympathetic for a second, but overall, it's rest, digest, right? If a lion came into the room all of a sudden, you would go into sympathetic. So sympathetic can override rest, digest, because if it has to do with your survival, it can take over. But most of the time, parasympathetic is in charge and sympathetic is in the background. Just in case you need me, I'm here for you. Okay? I'll kick it up a notch and we can run away or we can fight. But overall, rest, digest, parasympathetic is for the most part in charge of most of your daily activities. Okay? Let's see. And talking about neurons. So now we can go into the histology part of it. So what do these guys look like? So you guys have already done your histology for the most part for lab. So you guys have seen some of this, but now hopefully it'll make a little bit more sense. So neurons have big, huge cell bodies. So they're saying here is your neuron, and he has a whole bunch of things coming off of it. Do you guys know, or have you looked at the names of these? Do you guys know what those are? The dendrites. And then it'll have an axon hillock, which is this area here. This is a very important area that we'll talk more about in a minute, axon hillock. And then you have this projection that goes whatever direction it ends up going to. Do you guys know the name of that? It's an axon. And then it will have, later when we talked about like the synaptic knobs, remember those guys? Where we sent our neurotransmitter across the synaptic cleft to the motor end plate on a muscle. So the end of the axon hillock, or the end of the axon would have like the synaptic knob at the bottom that they're not showing in this particular picture here. So neuron, big cell, large cell body with a nucleus and a big nucleolus. So this part is the nucleus, and that other dot inside of it that looked like an eye is the nucleolus. The nucleolus is what makes our RNA, so ribosomal RNA. And what does ribosomal RNA mean to you? We're going to make a lot of protein. Exactly. So protein synthesis, and in this case, the protein that we're making in significant quantities is neurotransmitter. So what neurotransmitter did we already talk about with skeletal muscle? ACH, acetylcholine, right? That I can say properly today and I couldn't say properly the other day. ACH. What was another neurotransmitter that we mentioned that had to do with, was it smooth muscle? Yeah, do you guys remember? Norepinephrine. Okay, they had different actions, but those are all neurotransmitters, and that is going to be possible, and so that's why we have all this rRNA that's going to be made, because we need to have tons of neurotransmitter that we can release whenever we want to. Somebody might want to shut the door, sorry. So we have the neurons, and then we have a whole bunch of glial cells or neuroglia, okay? And those guys are our support cells, okay? So this, you could think of it like if the neuron was the surgeon in the OR, then all of the other people, the assistant doctor, the anesthesiologist, the nurses in there, the PAs, anybody else that's working in that room would all be the glial or neuroglial cells, the support staff, right? The ones that can jump in and help out, whatever the neuron needs help for. So that's what we're kind of talking about here. Glial cells, these support cells, make up 50% of the brain volume, okay? And so the neurons make up the other half, okay? Of the glial cells, there are 4 in the CNS and 2 in the PNS that we're going to talk about. Okay. So in the CNS, one of the ones, and they outnumber neurons by 10 to 1. The reason they outnumber them is the neuron is huge, right? And we said 50-50 on the volume of what makes up the CNS, but the support cells are all very tiny, because the neuron is huge and all these other guys are tiny. So you can have tons more of these. So there are 10 times more support cells to the one neuron that you have in here. And one of the cells of the CNS that's a support cell is an astrocyte. Astro means star, so it was given its name by the shape of it. So the astrocyte over here is the green one. So you can kind of think of it as kind of looking like a star shape there. And it's the most abundant glial cell in the CNS. So if you're not sure of anything else, it's probably an astrocyte up there, right? So if we look at the chart up here, it says astrocytes help form the blood-brain barrier. So if you look at this green star shape here, he has these projections that kind of come off, and notice, and they kind of can wrap around from the other side. And what do you guys think he is wrapped around here? This red thing. Take a wild guess. It's red and a tube. It probably has red blood cells in it. Let's call it a capillary. All right. I know your guys' mind is all on exam 3 material. I get it. All right. Maybe I won't ask questions. I'll just say stuff today. So basically what it's saying is it helps form the blood-brain barrier. So it has these projections that are kind of like little foot pods that are surrounding the whole capillary, so that things can't just come out of the capillary that are too big. So only small things like nutrients will be able to seep out of the capillary into the brain, but larger things would not be able to pass through there, and the astrocyte helps make that blood-brain barrier. Okay, we don't want viruses getting in here. We don't want bacteria getting there. Things happen sometimes, but we need to keep the CNS as protected as possible, and this is one of those protections, okay? It also says it regulates interstitial fluid composition. So basically, what it's talking about, interstitial, it means the fluid between one cell and another, so outside of the cells is interstitial fluid. And so it's regulating the ion content around the neuron. So everything here is designed to support the neurons, right? So for example, you guys learned that we could change what was going on with muscle physiology if we opened up certain gates and allowed certain ions to pass through. It then could cause a voltage change that then caused some other ion to get released and go through a gate, correct? So it's the same thing up in here. That's why I said if you learn muscle physiology, learning neurophysiology gets easier. So this ion content, we're talking about things like sodium, maybe potassium, potentially calcium, that kind of thing, that we're regulating and making sure that we have enough of what we need. So if we want to depolarize our cell and cause an action potential to happen, we're ready for it. So we're helping make sure that we keep this ion concentration at the right level for this area. It also provides support and organization to the CNS. So we don't have connective tissue up here. So this is like in place of connective tissue, these astrocytes help give support, some structure, scaffolding, if you will. Also, it says assists with neuronal development. So as a fetus, when you are still early development and your first things that's starting to develop is going to be things that have to do with the neural side of your body, these guys will secrete chemicals that regulate the connections between the neurons. So you'll learn in 228 that there are cells like in your white blood cells that can sniff out other chemicals that tell you there's damage somewhere. This is kind of the same thing. This is going to sniff out a chemical that says, oh, we need neural connections over here. So it's helping direct and say, come over here, let's grow over on this side now, let's do this over there. So it kind of can figure this stuff out when you're still a fetus. And then it also says replicates to occupy space of dying neurons. So if for some reason, unfortunately, you have a neuron that dies, gets damaged, and then dies, the neuron can't replicate. And so we'd have this gap in that area, right? As the cell atrophies and gets eaten up because now it's not a living cell anymore, astrocytes will fill in that space. Okay, so astrocytes, glial cells, are mitotic. They can divide. They can make more astrocytes and fill in that space from when a neuron has died and you have a gap. This cell will make another astrocyte and put that astrocyte there. So if you had scar tissue in the brain tissue, it's probably because of an astrocyte that filled in that spot, okay? Let's see, functions of ependymal cell, or microglia, sorry, let's go in order here. So we have microglia. Microglia are another glial cell of the CNS, and that's the third box down here for microglia. So it says phagocytic cells that move through the CNS. So phagocytic means that if you see something that doesn't belong there, see something, say something, right? If you see something that doesn't belong there, you would go over, this microglia would go there, and he's like, does surveillance. He kind of goes along and he checks things out and he says, hey, wait, you don't belong here, or you're debris. I'm going to phagocytize you, bring you into my cell, literally eat you and break you down because you don't belong there. So that's what microglia cells are. They surveil the area and they make sure only stuff that's supposed to be there is there, not funky other stuff, and get rid of the things that aren't supposed to be there. And it says it protects the CNS by engulfing infectious agents and other potential harmful substances. So kind of the same thing. If it sees like, let's say it was a neuron that had died, it might phagocytize parts of the neuron to break it down and get rid of it. So it can break it down that way too. And other infectious agents would be like bacteria, virus. It sees something that is not you, it doesn't belong in there, it's going to get rid of it. It's when things can't get rid of it that you have a problem, right? So you don't realize this stuff is going on all the time. It's always looking and searching and destroying this stuff. Let's see, what else do we have here? We have, next is ependymal cells. So that's the middle box here. So what do the ependymal cells do? They line the ventricles of the brain and central canal of the spinal cord. So here, sorry, was your microglia. See how tiny he is? This is a microglia there. The ependymal cells are these kind of columnar cells, and they actually will have little cilia sticking off. And this area here says it's the ventricle of the brain. So when you guys are looking at the ventricles of the sheep brain, you guys will see that to you it'll just look like a space. But in the living tissue, it had cerebral spinal fluid in those ventricles. And so ependymal cells have cilia, and they also help to circulate the cerebral spinal fluid. And in some areas, they may help make the cerebral spinal fluid. But think of it this way, as they help keep it moving around. So your cerebral spinal fluid does flow inside of your brain, okay? And around, it bathes your spinal cord as well. So, and you have a dura mater that helps keep it where it's supposed to be, so this doesn't leak out to the rest of your body. And if you've seen somebody that says, you know, it's bad if you have like clear fluid leaking out of the ear, right? Then they get worried that it might be your CSF if you've had trauma there. Not good to have CSF leak out, and you'll have headaches if that happens. It also doesn't feel good if you have too much fluid and it's not being drained off properly and stuff, okay? So let's see. So on ependymal, put has cilia. So those are little hair-like things, right? And the last cell that we have down there is oligodendrocytes. So these guys myelate and insulate the CNS axons. So oligodendrocytes are in the CNS. You're going to learn of another guy that's not in the CNS. This guy helps put the myelin sheath. So I'm going to erase this line on the axon. And for example, here is an oligodendrocyte. It has like a foot plate, and it put a myelin wrapper around there. And it actually made another one here. So and then look, it's connecting another neuron over here and puts a wrapper on it. And the myelin sheath does not connect. There's a gap in between them. Okay? So the myelin, this insulation that these oligodendrocytes are making and putting on there, they don't touch the next foot pod that put another one on, if you will. Okay? And we'll see in detail what this looks like in a moment. But this myelin that insulates it, you can think of it like electrical wire. We put insulation around it to protect it. So this is partly for protection, but it also actually allows, when we send a signal down the nerve, it makes it go faster. So it speeds up the delivery of like action potentials and things like that. So without the myelin, it won't go as fast. And there's a disease process we'll talk about later, multiple sclerosis, where these myelin sheaths get broken down and eaten away. And when you don't have that myelin there, then people have problems with their muscle control because the nerve isn't going to get the proper signal to the muscles on how to work right. Okay, so this myelin sheath is important. So that's the oligodendrocytes. That's this guy here. It says myelinates and insulates the CNS axons. It allows for faster action potential propagation, which just means speeding it along, propagation, along the axons in the CNS, okay? So those were the 4 glial cells of the CNS, astrocytes, microglia, ependymal, and oligodendrocytes. Now let's look at the 2 that we need to learn about in the PNS, or peripheral nervous system. So this is outside of the brain and spinal cord area. We have satellite cells, or neurolemmacytes. And the other name for neurolemmacytes is Schwann cells. So these 2 are glial cells for the PNS. All right, so satellite cells surround the neuron cell body. That's an N, neuron cell body. And of the ganglia of PNS, of ganglia, because we said ganglia and nerves are PNS, correct? So here it says functions of the satellite cell. Here is your satellite cell here, or an example of one. And this is his cell body right here, and there's his oh, shoot. Oh, nucleus, thank you, and nucleolus there. And then he has an axon, and in this case, he has an axon going this direction and this direction. And anybody want to take a guess as to what this might be here? It doesn't really look like a butterfly, but let's pretend that it does look like a butterfly there. Anybody want to take a guess? A cross section of your spinal cord? Okay, so it's out here which means it's not in the CNS. CNS is brain and spinal cord. This is PNS. This is where the nerve has exited, and actually you'll learn that this is a dorsal root ganglion, which is important later that you guys will learn the name of this. It's a little bulge. And then out here is where it will then exit the spinal column. So this dorsal root ganglion is actually outside of the CNS, but it hasn't exited the spinal column yet. But then it will become a peripheral nerve, thus the PNS, and go out to your hand or your foot or something like that, okay? So satellite cells surround the neuron cell body, located in the ganglia of the PNS. It says it electrically insulates PNS cell bodies. Let's see, and regulates nutrients and waste exchange for cell bodies in the ganglia. If we look at Schwann cell or neurolemmacyte, what does neurolemmacyte kind of sound similar to? Lemma. Sarcolemma? What was a sarcolemma? That was the cell membrane around the muscle cell, right? The plasma membrane, okay? So neurolemmacyte, so just like sarcolemma, cyte means cell, neurolemma means plasma membrane of the nervous tissue. So this forms the PNS myelin sheaths. So oligodendrocytes did the CNS myelin. This is PNS myelin, is a neurolemmacyte or Schwann cell. Okay. And so it says here for neurolemmacyte, it says it myelinates and insulates the PNS axons. So here is the neurolemmacyte right here. And again, it doesn't touch the next one that's here. It's wrapped all the way around the nerve there. Okay. And it allows for thus faster action potential propagation along an axon of the PNS. So same thing, anytime you see myelin, it speeds up the action potential and it insulates and protects the nerve, okay? So let's look at neurons, commonly called nerve cells. So it says there are some unique characteristics, and notice that a lot of these characteristics are the same thing that you had to learn about on the skeletal muscle or muscle in general. So you have excitability. It has to be able to respond to a stimulus. And just like muscle cells, and these guys, that stimulus can either be chemical or mechanical. What would be chemical? Chemical might be an ion concentration change. Okay? So if you all of a sudden flood your cell with sodium or calcium, and then you thus change the what do you call it? The positivity, negativity of the cell, because it's overall a negative cell, right? With the negative proteins lining the inside. So if you flood it with positive ions, it's a chemical. You have changed the positive or negativity of the cell. You allow for an action potential to be made, and you go from there, which we're going to learn more of those details in a second. But chemical would be like an ion gate that opens and allows ions to flood in. Chemical could also be a neurotransmitter, right? Yeah. Mechanical would be like an actual pressure change. So pressing my finger and deforming my finger, a mechanical change. Something squishing my finger is giving me the sensation that something is flattening. It doesn't hurt. I'm not getting a pain signal, but I'm getting a something is touching and squishing me feeling. Somebody pokes you in the shoulder. That would be a mechanical stimulus. Your brain interprets it and understands what it means. But those are all mechanical things. Something physically done could mechanically give you a stimulus. So the excitability. Conductivity is a voltage change moves along the plasma membrane, just like on a muscle cell. So we already learned about that. Secretion of neurotransmitter, and in this case it's from axonal endings, which we did see a little bit of when we talked about the muscle. These are the guys that are releasing acetylcholine across then the synaptic cleft that stimulated the motor end plate on the skeletal muscle cell, right? This is that. The secretion of neurotransmitter is that axonal ending, that synaptic knob that we talked about before. So it needs to function for a lifetime because we said our neurons don't divide, right? It dies, what replaces it? What glial cell replaced it if it dies? Astrocyte, exactly. They do not divide, thus the amitotic, which is why they have to function for a lifetime, because if it dies, it's gone, right? Exceptionally high metabolic rate, so it cannot survive more than a few minutes without oxygen. So they burn through their fuel quickly. So for example, in our muscle cells, we could store glucose that we could use for later. How did we store it in our muscle cells? Glycogen, okay? Our brain can't store glycogen up there. So we're in trouble. If we burn through the energy that we have, you need to bring me more so I can keep functioning, right? And remember, if you die, your muscle cells didn't know that you were dead yet, and the whole story about rigor mortis that we talked about. So they're still functioning until no more nutrients or no more oxygen or no more ATP was available, right? And then eventually it died off, but you were already dead, okay? So the brain, when it dies, it knows you died, but the rest of you still is functioning until it figures out that there's no more nutrients coming, we're not breathing anymore, I can't get my oxygen. These guys don't last long without oxygen. And when they get deprived of oxygen for too long, which doesn't take very long, then do you guys remember I said before that lysosomes, remember they're inside of your cells? What's inside of a lysosome? A whole bunch of enzymes that they're encapsulated in that vesicle so that they don't damage anything else. That vesicle might meet up with another vesicle, and then it dumps its enzyme contents in there, and it'll eat up like a, maybe you had, your phagocyte had a piece of mitochondria in there that was, you know, chewed up from before, and now you're going to finish chewing it up. It was already dead, this cell ate it, and now you're going to join with that vesicle, and you're going to get rid of it, right? So in this case, you lack oxygen, and all of a sudden these lysosomes up there, it's like they're suicide packets. They start bursting, and they release then their enzymes anywhere. They're not encapsulated anymore. And so then more things get damaged. So if somebody has a heart attack, what they're doing is they're trying to keep oxygen in you long enough until they might be able to revive and get the heart to start again. Okay, it's the oxygen side of it that they're concerned about. If you fell into the cold river and you essentially die, heart stops, and they get you out, everything has slowed down, so they might buy you a little time because you were so cold that it might just give you enough time to start the heart up again where you didn't lose enough oxygen where those enzymes started popping from the lysosomes. It didn't pop out and start doing its suicide packet. So keep that in mind. That's what we're talking about on high metabolic rate. Lysosome starts doing auto-destruction. So again, you don't have glycogen storage. You don't have myoglobin that the muscles had for where you could rob a little bit of myoglobin oxygen until you got more hemoglobin oxygen coming to you when we talked about fast glycolytic and slow oxidative and fast oxidative energy for last exam. Review that stuff. Yes, do so. Okay, neuron cell body. Soma, we said meant body, right? So it says the biosynthetic center of a neuron. And we have rough ER, RER, which it says chromatophilic substance or anisal body. So chromatophilic, philic versus phobic. Phobic means you're afraid of, so philic means you like it or you love it. Chrome means color. So these are color-loving substance. So anisal body is going to look very dark. So kind of dark staining when you see that on, if you guys looked at that under the histology for lab. Okay. So Nissl bodies, because it's rough ER, what does rough endoplasmic reticulum mean? It means you have ribosomes. Ribosomes mean protein synthesis, right? So if you see Nissl bodies, you see rough ER, you know that we're talking about protein synthesis. If we look at neurofibrils, so these are bundles of intermediate filaments. They maintain the cell body shape. So I know it doesn't quite make the same sense as myofibril, so you'll just have to memorize this as being a little different. So neurofibrils are bundles of intermediate kind of means like in between, intermediate filaments, which maintain cell body shape. Neuron processes, so these are extensions off the cell body. So you guys already kind of mentioned dendrites. So these are on the receptive end of the neuron, meaning that this is where they're getting information at the dendrite or receptive end, and then thus it'll send it down the axonal end to either talk to another neuron or send the information onto another area of the brain, right? So, so dendrite receptive area conveys impulses to the cell body. And so this is talking about the input side, info coming in. And these will be hundreds of dendrites per cell. Okay, so you'll have, think of it as like crazy hair day. Tons of little dendrites hanging off the cell body, right? So hundreds of dendrites per cell. If you look at the axon end, so this conducts the impulse from the cell body to another location. So there's just one per cell typically. It's referred to as nerve fiber or just fiber. So when I'm not talking about skeletal muscle, now that we're talking about nervous tissue, if you hear me say the word fiber, it doesn't mean skeletal muscle as in muscle fiber. It now means, if we say fiber, it just means nerve fiber. It means neuron, the axon end. Okay? And so this is the output side, right? Input dendrite, output axon. Here's that term that I said we'd come back to, axon hillock. So this is a cone-shaped part of the neuron cell body where the axon attaches. So for example, here we had crazy hair going on, right? Here was your big cell body. There's all your dendrites hanging off doing their thing. And the axon hillock actually has this cone shape here. So this is the axon hillock coming in this direction, okay? And so that's the cone-shaped part of the neuron cell body where the axon attaches. So that means anything down this way is the axon, right? And we said there's just one per cell on that. And what do I want to tell you about that? So this is an important landmark on neural physiology. So that's why we're specifically talking about an axon hillock. We'll come back to that. And it also has axon collaterals. So these are branches off the main axon. So he might have a branch near his end like this. Okay, so there's one axon, but then there can be axonal collaterals. And then you can also have terminal branches, which means it's found, if you guys think of the word terminal, like the end of something, terminal bus station, end of the bus station, no more bus stops after that. So terminal branches are found at the end of an axon or collateral. So this would be like a terminal area, terminal area, terminal area, that kind of thing. So these are tiny branches and up to about 10,000 of them. And they can also be called telodendria. Telodendria. So telo means end or last, so that's just the same, the end of the axon, essentially. Synaptic knobs, you guys were already familiar with when we talked about skeletal muscle physiology. So the synaptic knobs were on the neuron that was communicating to the muscle. So it's the knob-like ends of the terminal branches where the synaptic vesicles are stored. And what's stored in our synaptic vesicles? Neurotransmitter, ACH, or it could be, if you're talking about something else, it could be norepinephrine or something else. But mainly we're talking about ACH as the neurotransmitter for skeletal muscle, correct? And so your synaptic knobs would be, you know, like you'd have like that little knobby there, and it would have the little tiny vesicles, and we had the big blown up picture, and then you had your muscle, your skeletal muscle there that had the motor end plate on it, that this would put neurotransmitter ACH across the synaptic cleft. So functional characteristics. So this is functional characteristics of the axons. They conduct impulses from point A to point B. So impulse conducts impulses from point A to B, okay? So axonal transport. So this is the main intracellular transport mechanism. So you can think of it kind of like a railroad system, and this is actually bidirectional. So it can go in both directions, okay? This is kind of unique. So it's a system of microtubules from the nucleus to the synaptic knobs. It's bidirectional, so it goes either way, which is good for a lot of things and not so good for some things. And it carries neurotransmitter, NT for neurotransmitter, mitochondria from the soma to synaptic knobs. And the other thing is, this is where I say it can not always be good. Some viruses use this mechanism, this bidirectionality, and the microtubule system. So some viruses use this mechanism to move within the neuron. So for example, polio, herpes, okay? You guys know like chickenpox is the herpes virus. Also later on in life you might have shingles, which is, you can't get shingles without not having had chickenpox when you were younger, okay? Some people may not get chickenpox when they're a little kid. Now we vaccinate for chickenpox, so you might, I'm still waiting to see what the outlook is on how many people get shingles who've had the chickenpox virus injection. I'm curious. But basically, the herpes virus ends up hiding out in your dorsal nerve root ganglion and revisits you sometime later on down the road when you're under a lot of stress. And it uses this mechanism of our axonal transport against you, essentially. So good to know, but not always fun. The rest of it is a good reason why we have it. We'll talk more about herpes later. The myelin sheath. So we said this protects and insulates and thus increases the speed of impulses. So if we're talking about PNS myelin, who makes the PNS myelin? The Schwann cell or neuroleukocyte does, right? Who makes it in the CNS? Oligodendrocyte. Okay, so right now we're just talking about PNS for a second. So Schwann cell or neuroleukocyte wraps itself tightly around an axon. So what they're trying to show you here is how it forms and how it actually wraps itself around. So this is one cell. So you're seeing the one nucleus, and here's your axon of the nerve, the yellow tube that's there. And this cell will first wrap around the nerve, and then he starts to wrap himself around. So there's the one nucleus, and he's going to continue to keep going around and around. Okay, and see now it looks like the rings of a tree on this picture. So many, many times around that it's wrapped around. The wrapping is its plasma membrane, okay? And so it's squishing, like it has like the little bump there where the nucleus is and where a little bit of its cytoplasm is. So there's not a whole lot of cytoplasm on these inner rings. It's kind of squished it out as it kept rolling around the nerve. So you have one nerve and it kind of gets on it and then just wraps itself around and around and around and around. And the more times it wraps around, the better, because more insulation. And then it'll be a different neuroleukocyte or Schwann cell that'll do the next myelin wrapping next to it. Okay, so as you move down your nerve, let's say here's our nerve, you would have one neurolemmacyte that wrapped itself around a whole bunch of times. Then you'll have a little bit of a gap. Then you'll have another one that wrapped itself around a whole bunch of times. Then you'll have another one. So none of these guys are touching each other, but they just keep going on down. Another one will attach and keep wrapping around until it's done wrapping. So that's what happens. So let's see, what do I want to tell you about that? So the bulge, again, is just the cytoplasm that contains the nucleus called the neurolemma. So PNS myelin defined. So it's a tight wrappings of a Schwann cell membrane around an axon without the cell nucleus or cytoplasm. So all the wrapping, remember this is one cell, so it only has one little spot that'll contain that cytoplasm that got mushed into one little spot with the nucleus. Everything else, it's kind of been squished out to the side because this is just one cell that wrapped and wrapped and wrapped. Okay, the neurolemma is the outermost remainder of the Schwann cell. So that's why they're saying that one little area with the nucleus is the neurolemma. Okay, and so that was for the PNS. Now let's look at the CNS one. CNS, I told you, was the oligodendrocyte, performs a similar function, but notice how he kind of, think oligo, think like octopus, right? Multiple projections sticking off. So this is one oligodendrocyte with one thing that wrapped around and caused his myelin sheath on that part of a nerve. He has a second one not touching this one that wrapped around there. And he has another projection that went to a whole different neuron and wrapped around there, and so on and so forth. Then you have another oligodendrocyte that he wrapped around here, and to this guy, and to here, and maybe down to this guy over there. Can you see all the many, many connections that are going on there, right? So an oligodendrocyte performs a similar function, but for the CNS neurons, okay? Now, in between those little spaces that I said, remember they don't touch in between these? And on my little hand drawing here, these little spaces in between here, those are what are called node of Ranvier, okay? So a node of Ranvier is neurofibril nodes, short sections of naked axon between the myelin wrappings. They're called the same thing whether you're talking about the CNS space or the PNS space between these myelin wrappings. So that's why it's saying it's in between there and in between here, in between here. Those are all nodes of Ranvier. Okay, so node of Ranvier, what else do I want to tell you? So Schwann cell or oligodendrocyte. So Schwann cell or oligodendrocyte, depending on whether you're talking CNS or PNS, right? Do not touch one another. Okay, and what did, so I already told you that the myelin will speed up how fast you can send an action potential down the neuron. These node of Ranviers, it's like where you get to skip from myelin sheath to myelin sheath. So this also, again, will speed up the impulse speed. So increase impulse speed. Okay, so from neuron cell body down the axon, this will also help speed that up. Now, we do have unmyelinated neurons, so neurons that don't have this neurolemmacyte wrapping around it, right, or oligodendrocyte. And so an unmyelinated neuron, okay, can be in the CNS and PNS, but Schwann and oligodendrocytes are associated differently with the axon. So instead of wrapping it around and causing that like tree ring shape where it spins around it, okay, causing the myelin sheath, they will sit on it a little different. So here we have 1, 2, 3, 4, like 5 different axons. Now they don't have myelin wrapped around it, but the neurolemmacyte, the Schwann or the oligodendrocyte, it's kind of like here's one cell, and you know, like people that go and get their toes done, like a pedicure, and they stick the spacers in between the toes so the toes don't rub and get your toenail polish on another spot. That's kind of like what the unmyelinated Schwann or oligodendrocyte is doing. It's not wrapping around the toe or the axon. It's keeping the axon spread apart so that way we don't cross our wires, okay? It kind of keeps things in line and it won't cause any problems with crossing the wires. So no tight membrane wrappings, no myelin with this. Most of our pain fibers are unmyelinated. And so they're not 100% sure why pain fibers may be mostly unmyelinated, but maybe it's so that we can have a delayed pain response. So maybe something mild wouldn't cause you to, jump out of the car all of a sudden. I don't know, when you're driving down the road at 60 miles an hour, that wouldn't be a good idea, right? So maybe it delays it a little bit, allows your brain to think about it before it decides a response, potentially. All right, so let's see on this picture. So these terms down here, nerve, tract, gray matter, and white matter, you're going to have to just memorize these guys. But the term nerve is a bundle of axons in the PNS versus a tract are going to be a bundle of axons, but in the CNS. Okay, so again, nerve would be easy to remember because we said only brain and spinal cord were CNS. So if we say nerve, you automatically associate it with PNS. So think nerve is a bundle of axons in the PNS. Then you can remember tract. When you guys are looking at the brain tissue, you'll be able to know that tract thus has to go with CNS. Also with CNS, you have gray matter, which the CNS neuron cell bodies are in the gray matter. That's what makes them termed gray matter. White matter are the axons from those cell bodies from the gray matter. So when you guys are looking at the brain dissection this week, you will be able to see gray versus white matter, and now you'll know that in gray it's CNS neuron cell bodies, and their axons make up the white matter part of it. What else do I want to tell you? Let's see. So again, white matter, gray matter, you're talking about CNS. Let's see. The reason white matter is white, so we said white matter is the axons, right? And this is similar to a tract, but it's a little different. And if the axons have myelin wrappings around it, what makes up our plasma membrane, the majority component of a plasma membrane? Because remember, we're talking neurolemma site here, right? Neurolemma, phospholipid bilayer, right? So phospholipids are white because the majority of that myelin wrapping is phospholipids. So it's going to be looking white. So think layer of phospholipids, myelin. There's not a bunch of other stuff in it because remember all the cytoplasm was squished out to where the nucleus is on one little end, right? So there are three types of neurons that we're going to look at, multi, bi, or unipolar neurons. So let's look at the classification of structure. So all have an axon, but we have to look at the dendritic end to kind of be able to decide who goes where. So multipolar, bipolar, or unipolar. Bipolar is not referring to a medical condition. Just kidding. Okay. So structural component. If you guys look at multipolar, multi means many. Look at how many dendrites are on multi versus bi or unipolar. So this is totally crazy hair guy, right? Tons of dendrites hanging off the cell body. So multiple processes extend directly from the cell body, typically many dendrites with one axon, and this is the most common type of neuron. This is what you're going to see most often, okay? And all motor neurons are multipolar, and most interneurons, inter means a neuron that's going to communicate in between other neurons, most of those are also going to be multipolar. So can you see how we've pretty much covered our bases why almost everything is going to be a multipolar neuron, okay? And this means, remember the dendritic side is the input side, so it means many going in or much information coming in and only one going out, right? Because what is out? Out is the axonal end, okay? If we look at the bipolar neuron, so that is a single axon and a single dendrite. So bipolar, can you see the cell bodies in the middle and you have one dendrite end, one axon end? Okay, so this is saying if there's one dendrite, how many is coming in? One in and one out, right? One on the input side, one on the output side. Okay, so it says two processes extend directly from the center of the body, one dendrite and one axon, relatively uncommon. So we don't see a lot of bipolar neurons. Where you're going to see a lot of bipolar is going to be with our special senses, which we start 228 with special senses. So this is special senses. These are rare, so we really only see them there. And an example of our special senses would be for our vision and smell, for example. So vision and smell. Okay. If we look at unipolar, so this is a single short process that extends from the cell body. So these are for sensory neurons only. And if you think about it, your sensory input is going to be very, very specific. You actually have different nerves that will tell your brain whether you felt something hot versus cold. You have a different nerve that can tell you whether you are touching something hard versus soft. Something else that tells you whether you have pressure against you. Something else that's, so just like how enzymes were very specific with what it could attach to, these guys are very specific in what they can sense. Okay, so these are sensory neurons only. And this is going to be peripheral and central axons. So instead of, so this has a unique shape to it. Here's your cell body, and it only has one projection that comes up, and then there's a debate on whether they think one side's really dendritic and one side is really axonal. So Sherry likes to say this is where they took the two and wrapped a ponytail on it, and then, oh, I don't have a ponytail today, I have a bun again. So it's like having your ponytail sticking out on both sides, but it had one area that it came off the cell body, off the head, okay? So instead of using the term dendrite versus axon, you'll hear them say peripheral process and central process as the two arms instead of dendrite and axon. Okay, so basically not everybody agrees with this terminology. Some say that the part leading to the cell body is the dendrite and the part leading away, because you know, must be the axon side of it. Others would say that the entire structure is an axon because myelin occurs along its length. And myelin is only on axons. And this has myelin on both sides. So that's why some people say, well, it's got to be axons on both sides. So we'll call one side peripheral and one side the central process. So we're going to call it an axon with a peripheral and central process for our purposes so that we keep intact with the book. So peripheral and central axons uni means one, right? And so one connection to the cell body. So where we see this, you actually just saw this a moment ago. If I scroll up to, back to this picture up here, this guy right here. Okay, he was a unipolar neuron. Notice there was myelin on both sides, right? And we see this in our ganglion, and that was a dorsal root ganglion is when we were talking about that guy. So I'll erase all the extra writing I saw put there so you guys can see that better. But see how he has one process coming down from the cell body, and then he has a projection that goes in either direction, okay? So that's what we're talking about. So I'm going to go back down to where we are in the notes. Don't get dizzy. All right, so unipolar, think ganglion. Okay. Functional classification. So according to the direction of the impulse. So here we're coming back to that terminology I introduced at the beginning where we have sensory is afferent. Afferent is going to the CNS. It makes sense that sensory, we take information from something that's going on versus visceral versus body, right? That we're going to send up to the brain, and our brain's going to decide what to do with that information. What does that info mean? So impulse direction is toward the CNS, afferent, ascending, okay? input side. All right. Cell bodies are located in clumps called ganglia in the PNS. So we've seen that, right? Ganglia, dorsal root ganglia we talked about. All sensory neurons are unipolar, right? Because we said the ganglia are the unipolar. These are our unique guys. So all sensory neurons are unipolar. They typically process a long peripheral process. Okay, so for example, if you were to say like the sciatic nerve, one, we said nerve, so we know that this is PNS, correct? Two, we said that the cell bodies are located in the ganglia of the PNS, so we're still talking PNS here also. Also, the peripheral process is really long, we said. So if you think about the sciatic nerve, if you're probably taller than me, it might be 1 meter long, okay? So it's going, your dorsal root ganglia, the cell body is right next to your spinal cord. It just exited the spinal cord. That's where the cell body is. And it's one of its axonal processes goes all the way down to your big toe, okay? And what else do I want to tell you? So ganglia are just outside the vertebral column. I said that. Good, good, good. All right, so that's the sensory part. If we are calling it a motor classification, motor neuron. So this is efferent. Efferent meant exit. So this is going away from the brain, away from CNS. So efferent, impulse direction is away from the CNS. This is our output side, correct? And so what that means is, let's see, it's going to the effector is another term that we can say, right? To effector. And the effector might be, if it's somatic, it's going to be your skeletal muscle. If it's autonomic, it's going to be cardiac, smooth muscle, or glandular, okay? So, is this also correct to say that the direction of the impulse is toward the PNS? We're sending the information from the brain to the PNS. Does that make sense? So then yes, okay? So motor cell bodies are actually in the CNS, but where is its projection? Outside of the CNS, correct? So, and then all motor neurons are multipolar, and we said that earlier in that chart. All motor neurons are multipolar. Most interneurons are going to be multipolar, okay? And so interneurons, another name for them are association neurons, connect the sensory neuron to the motor neuron. I wonder where these guys are located. I'm glad you asked. Let's look at that. So the cell bodies are going to be in the CNS. They are all multipolar, okay, because we said interneurons are multipolar, most of them. 99% of all neurons are interneurons, okay? So you guys hopefully already looked at the difference between a Purkinje and a pyramidal cell, okay? Pyramidal neuron and Purkinje neuron. So pyramidal is found, is it in the cerebral cortex or the cerebellar cortex? Pyramidal are in the cerebral cortex. So pyramidal had like the pyramid shape to it. So pyramidal and cerebral, the cerebrum, right? Cerebral cortex versus Purkinje, they are going to be in the cerebellar cortex. Okay. So you can think of Sherry's little mnemonic for this is that cerebellar has like two L's in it, so it's like the legs, and you need your cerebellum for balance and walking straight so you don't tip over or look like a drunk person. Okay, there's actually problems where people have damage in the cerebellar cortex where they look like they're drunk, but they're not inebriated. Okay, so you just have to remember Purkinje is in the cerebellar. Let's see, and then we're going to end up talking about simple to complex circuits. So let's see if I can say that really quickly before we end this section. All right, so simple to complex. So simple would be like a reflex, which we'll talk into a lot more detail about a reflex. You know, when the doctor does like the reflex hammer on your knee and watches what happens, you don't think about it, and it allows the sensation actually goes and causes the leg to kick out without talking to your brain about it. It doesn't ask for permission. Can I kick my leg? Okay, so that's a reflex. That's simple. Complex would be like what you guys are doing for exam 3 right now. You are studying difficult material and you're trying to make information make sense. That's complex thought processes. Okay, so that's what this has to do with. So this is where we make decisions between sensory in and motor out. And we said earlier that some people older people actually have less neurons, but kids have more, but older people have more complex circuitry. So kids have more neurons. We actually, as we get older, make more connections and have less neurons. But we make much more complicated thought processes than what kids will make, right? Does that make sense? Okay. So, like, kids can't do algebra. I mean, maybe Genius Kid could. Okay, I won't go there. But little kids aren't making difficult thought processes. They're not studying A&P like what you guys are trying to do. Their thought processes is more basic and simple. And then as they get older and have more experience, they make other interneuron connections, okay, that associations between things, so then they can make more difficult, thoughtful, complex thought. So a person that has more complex circuitry. So not the number of neurons, but the sophistication of the wiring, essentially, is what we do, how we turn it on and off. So if you want to think about like a 3-way light switch, your neurons that make these extra connections, it's like a 10,000-way light switch. You click on one and it's making information switch on and off other things really, really fast. Okay, because again, if you think about it, you have this neuron with its dendrites here. Those dendrites are talking to other axons and other information over there, and this guy and this guy and this guy is, and then his axon is coming down and talking to somebody else. And remember, he has maybe terminal branches, so he's talking to other things. So that's why I say it's like a 10,000-way light switch when you make these interneuron connections. Okay, and what else do I want to tell you? So learning as a kid, it's like only 500 connections. As a teen, you might have 1,000 connections. By the time you're in college, it's exponential. And so a 5-year-old has more neurons, but you have more connections. When you're learning, you are building these connections. So also like a weightlifter, a 1-pound weight, would you build muscle if you just lifted a 1-pound weight? Not likely, right? So the more difficult weight to achieve that, you'd have to lift heavier weights in order to do it. If you take a class that doesn't stress you to the point of your head hurting, like this class, are you really accomplishing or learning some stuff in some of your easier classes? No, this one is hurting your brain. You guys are not liking me very much right now, right before an exam. I know that, but you guys already think different now than you did the first day you walked into class. You guys have made more connections of learning pathways and how you had to memorize stuff, and you do things and approach things differently now than you even did before, okay? So let's see, dementia, for example, what's happening to the neural connections when somebody has dementia, do you think? Do you think they're all there? Not so much. They're losing those connections. So you can see that actually under a microscope. You can see the loss of connections, okay? So they're becoming more childlike. Have you talked or had interactions with somebody who has dementia? And they tend to be more childlike in their thought process and how they talk and remember things and do things than they did. And this is why. So think about it that way when you're trying to think about all these interconnections that we have and how we work with it and stuff. So let's see, what else do I want to tell you? So that's the whole idea of the simple to complex with interneuron associations. When we come back, I'm not sure yet if we'll record a lecture, even though you guys are taking your exam on Thursday. We will pick up again at membrane potentials, and this is going to talk about how do we get that neuron to fire and to tell that muscle cell that you guys are being tested on for muscle physiology how to do it and when to do it, okay? So let's stop the lecture there.